Well, we looked at the procedure of Christian Graham. Now let us look at the interpretations that were later given to this observation. It was later observed that bacterial walls are of two types, and the two types are mainly due to the difference in thickness of this part here, this purple part. Look here, if you look here, this purple part is very thin, is very thick. And if you look at the diagram below, this purple part is very thin. In this diagram, it looks like, please remember, this is the cell membrane which we have just discussed, the phospholipid bilayer, and this is the wall. It is just one solid structure. But here, if this is the cell membrane, we have the wall and what looks like a membrane again on top of it. Now, the thing we have on top here is a membrane, but we don't call it a cell membrane. We call it an outer membrane. It is different from this membrane by composition. Okay, I'll come back to that. But for now, let us think about this purple part, the thick part of the wall. This part of the wall is what we call PDG. It is what in plants is the cell wall, the structure made of cellulose, microfibrils, macrofibrils, until we have the rigid wall. But here, the wall, as we can see from this diagram, is a complication of a lot of things. But what Christian Graham was observing was the peptidoglycan part. Peptidoglycan, which is shortened to PDG, and again, I'll caution you, know it first as PDG before, know it first as peptidoglycan before you refer to it as PDG. Now, in gram-positive walls, we have a thicker layer of PDG, and in gram-negative walls, we have a very small layer of PDG. Now, I'll come back to these diagrams. There's a simple diagram which I want to show you, which I think you can reproduce. Please note, I've taught this course for quite a long time. The library is full of tests and past exams. As you start your course, go and collect all those tests and all those exams and see when you finish a chapter or a topic to see if you can answer these questions. If you can't, that means you have a problem and you must ask. Okay, let me go to a simple diagram which I made myself, and I'll tell you why I made it. Okay, this is the diagram. Please note the wall starts here. This is the cell membrane, the one which is in blue, and the wall starts here to the end. The reason why I have this diagram is because when I ask you to draw a gram-positive wall and a gram-negative wall, I don't want you to go into the complications that students sometimes get into. In In addition, this diagram has got dimensions here. It tells you how thick each part is. Okay, this is the gram-positive wall, and this is the gram-negative wall. This is peptidoglycan, and in the gram-positive wall, it is 15 to 80 nanometers thick. Here, this is the only part which is peptidoglycan. This, I deliberately colored it or shaded it darkish for you to be aware that it is part of. Okay. So the reason why I like this is because as I told you, here we have the peptidoglycan, which is 15 to 80 nanometers, and here it is only 2 nanometers. Then up here we have the outer membrane, and the outer membrane is 8 nanometers, similar to the cell membrane, 8 nanometers. Now, here and here, above and below, uh, above and below this peptidoglycan, we have a space. Please, when I say space, I don't mean empty. It just means an allocation. We have an area which is full of enzymes and other um, fluid bound elements. So this is not empty. Otherwise, this would collapse onto that, and that would collapse onto this. So much as we call this the periplasmic space, it's below and above the 2 nanometer of peptidoglycan. It is not empty. Then on the outside, we have capsule. Capsule, I said, when it comes to size, it is variable because it depends on the environment the cell is in. Does it need excess protection or little protection? So slime or capsule is of a variable size. And then I have shown you flagella. This is a flagellum and that is a flagellum. And in each, please note, the flagella have got these things here which we call rings. Remember, flagella are for motion, not locomotion, but motility, ability to move. Normally, when you say locomotion, you are implying existence of a skeleton. So we rather call it motility. So here, this is the flagellum, and it has 4 rings, an L ring, a P ring, an S ring, which is above the membrane, and an M ring, which is in the membrane. This is in the gram negative. In the gram positive, there are only 2 rings, one in the peptidoglycan and one in the membrane. But here we have 4, one in the outer membrane, one in the peptidoglycan, one above the membrane, and one in the. So we have the LPSM rings. Now, I've also given you the dimensions. A flagellum could be 12 to 18 nanometers thick, while a fimbria, depending on whether it's reproducing, the organism is reproducing or not, it can be 4 nanometers up to 35 nanometers. So please, this is your key to the colors that have been used. Capsule is brown, cytoplasm is red. This is to show you where this, here I've shown you the mesosome. Look at the rest of the cell. At the end of this, you should be able to know everything that we have covered. Let me show you another slide that will show you the actual rings. Okay, this is a gram negative wall, and here are the rings, and this is the flagellum, and here are the rings. These diagrams you will find in the booklet that I hope you will purchase. Please, this is the LPSM ring. Please, when you register, you also have to write down whether or not you are willing to buy the 2 booklets I told you about. And the reason why you should write down your name is because we do not want to waste our supplies buying and making booklets that nobody will want. Okay. So the M ring is in the cell membrane. So So remember, M, cell membrane. S ring, just have to remember it's on the membrane. Okay. I told you about the S ring. I said it's on the cell membrane. S for cell, if you're not thinking of cell as C, but as a cell. Okay. P, it is in the peptidoglycan. L is in the lipopolysaccharide. Lipopolysaccharide is part of the outer membrane. And I told you that the periplasmic space is not empty. It contains certain enzymes, proteins, and oligosaccharides. There are certain chemicals that we are going to meet. Now that I'm on this slide, let me just talk about them. There is a chemical called terechoic acid, and terechoic acid is made up of glycerol bonded to alanine. It's glycerol bonded to alanine and a phosphate. And we have lipopolysaccharide, which you will see on the beautiful diagrams. Lipopolysaccharide has this O-specific chain, which is hydrophilic, and it has a core of oligosaccharide and lipid A, which is hydrophobic. So on the outside, it's hydrophilic, inside hydrophobic, and it is lipopolysaccharide. And once you are familiar with it, just call it LPS. The lipid A, which you see here, is toxic to animals, causing in the animals it will cause endotoxic shock. This lipid A binds firmly to the cell surfaces, and it is released when the cell lyses. Remember, a bacterium is also a living organism. It has to protect itself from things that will attack it, animals that will take it into their systems. So if the bacterium is in the cell, like you might have a fever, whatever, animals will go into shock because the LPS has this lipid A. Now let's find where all these compounds are. I'll come back to that shortly. Let's go back to the beautiful diagram at which we left with all those rods on it. Okay, now the reason why I want you to look at these diagrams is to understand how PDG or peptidoglycan is different from cellulose. I told you that cellulose is made of repeating units of glucose. This, if you look at, let's take one rod. If you look at one rod, you see that it is light, it is dark, it is light, it is dark, etc. The light part is nag, the dark part is nam. Nag, you'll call it nag after you have learned it is N-acetyl glucosamine. Nam is N-acetyl muramic acid. So there's no complication here because all you have to remember is that the G is for the glucosamine and the M is for the muramic acid. But to make a rod, we have repeating units. We have nam, nag, and this is what makes the peptidoglycan. This is a very rigid or solid structure. However, these rods would be collapsing into each other. They are held together. They are held together, but by these beads which you see here. If you look properly, that these green beads here, and across the beads are blue. These are making sure that the distance between the rods side by side is held in place. The distance between the rods going down is also held in place. I will show you a diagram where this becomes a bit clearer. That's why we have so many diagrams. Now, after studying the wall for so long, let's look at its functions. I told you that when something is on the outside, the first function you shouldn't even study is the fact that it will protect. It will protect the cell from damage. When a cell is damaged such that it bursts, we call it lysis. However, if a cell just bursts a little bit such that you have a small bump coming out, this localized imbibition of water is called plasmoptosis. 2, the cell also acts in the regulation of the flow of certain divalent cations. I've told you which ones. Then the cell wall is also a molecular sieve. Things just don't go and come as they please. They are blocked by their size. Then we have the porine trimers, which are present in the gram negative, and these are also used for free passage of certain small hydrophilic molecules. And the cell wall also contains transport systems, especially in that periplasmic space. Okay. Now, outside of the wall, we have the capsule. Sometimes you find they say we have slime. We call it capsule when it is relatively rigid, and we call it slime when it is loose, or viscoid or colloidal. Okay, but what is the point of this external protection? This capsule is mainly made up of water, but there are some solids, about 2% of solids. But for bacteria that always have this capsule, it is involved in pathogenicity or virulence. Please note, pathogenicity is not the same as virulence. Pathogenicity is the ability to cause disease. Virulence is the intensity of the disease. However, the presence of capsule can make a bacterium disease-causing or non-disease causing. Please keep this in your mind as we go into sexual reproduction and when we talk about transformation. You will understand what I'm talking about with pathogenicity. So the functions of the capsule, again, because it is outside, it will be protection. And the other protection is ingestion by phagocytes. Phagocytes are cells of the host, or phagocytes are cells of the immune system that help in cleaning your system. Now, you want to get rid of the bacterium wants to stay in the system. So the bacteria has also its own ammunition. It has the capsule to make it difficult for the cell to swallow it. So the other thing is Streptococcus pyogenes has in the capsule a protein which we call the M protein. We use this. It has M protein and hyaluronic acid. We use this to identify when you identify it in the lab. Not our lab, of course, in medical labs. And the capsule of Bacillus anthracis has D-glutamic acid, which is also used to identify it. But please note, the bacterium is not keeping these things for you to identify. It is keeping it for other functions, but you are now using that to identify it. Okay. It's just like saying, if for example you have something wrong with your face, you are not putting it there so that people can know you. It's just because you have that, then people know it is you. So that is what I'm trying to say. Um, so the capsule is important in immunological reactions. That means in this antigen antibody reactions which you study in immunology, the capsule is important because it is the one that reminds the cells of the host whether this bacterium has been there before or if it is a first visitor. So the memory of a certain bacterium might just be a memory of its capsule. Then your system knows that this bacterium has been here and this is what I have to release to stop it from multiplying. The flagella, as I told you, are agents of motility. Now, why should things be motile? But before I get to that, please note that the flagella, you saw the wheels, the flagella will move. And I know in your mind you have told yourself that if something moves, it can't be a plant. Okay? There are things that are at the border there. Some would call a plant and justify, some would call an animal and justify, some would call a microbe and justify. The important thing is that as long as you know what you're talking about. So the flagella make the bacterium motile, and it is this flagellum is made up of a protein which we call flagellin. Um, This flagellin is similar to myosin, which is the protein that makes up your muscles. Now, why should a bacterium have a flagellum? Okay, just think of yourself. Why do you move? One, you move because you are hungry, you want to go and get food. If you stay in one place, you'll exhaust your supply. So they also move to increase their nutrient uptake. Two, you also move because you want to run away from a toxic or dangerous situation. So any environment that is not conducive, we say is inconducive, you run away from. So the bacterium also wants to run away from situations that are dangerous to it. Three, the bacterium uses that just like how you would use a screwdriver to nail, to push a nail into a wall or into a frame. It penetrates certain barriers by that spiral motion. Okay. The fimbria, the fimbria or the pili are the organs that are used by the bacterium for adhesion, and at the same time, the bacterium will use the same organ to transfer a nucleic acid during reproduction. And uh, lastly, it will be an entry point for certain viruses that come in with foreign nucleic acid into the cell. Sometimes this is good for the bacterium, sometimes this is bad. So generally we can say fimbria are used in reproduction. Think of yourself when you are reproducing. You can't reproduce with somebody when you're standing in one end of the room and they're standing by the door. You need to be together, not only together, but very close to each other. Actually, whether you like it or not, you have to hold each other in place for some time. That's what bacteria uses its fimbria for, to hold on to its partner for a while. We'll see this during conjugation. And during another process, during the same conjugation, you will realize that they're not only holding each other together, they are also using this passage or this communion between them to pass the nucleic acid over. Assume as your male partner passes nucleic acid over to you using a passage, that passage, instead of being whatever you know it to be, just think of it as the arms that are holding that you are holding each other with. It uses the same arms to pass the nucleic acid over. When it comes to this viruses, how they use this pili as receptor sites, let's wait until we come to transduction. Now, there's something I want to tell you about reproduction, and this is the fact that reproduction, especially sexual reproduction, is not there for fun, for you to enjoy. Sexual reproduction is there for you to fulfill God's instruction of increase and multiply, but also bring about new organisms. So in sexual reproduction, you bring a new organism that is different from both of you. And that difference from both of you is what makes it very important, because we always believe that the new that we are bringing is normally improved and better than what you are. Okay, but when we are now going to move on to reproduction, but we will start with asexual reproduction, and when we finish asexual reproduction, we'll go on to sexual reproduction. But please keep what I've said about the pili in mind. This should not be a problem for you. Okay.